So I turn to the first speaker and say that it's with great pleasure that I introduce Arieh Warshel. And I don't know whether this system here will work. Can my assistant tell me what to do? I don't know. Who is the gentleman that operates this? Ah, good. So I met Arieh several years ago in Sheffield when he was the centenary lecturer of the Royal Society and not then yet a Nobel Prize winner. And I began to read his work. We had narrowly missed overlapping in Cambridge in the Laboratory for Molecular Biology, which I stopped collaborating with about 3 years before he arrived to work with Michael Levitt. And Michael gave him a new inspiration, which he carried forward and led him 3 years ago to the Nobel Prize. I want simply to say that, if the slide, can you work it? Mon Dieu. C'est impossible. So, forget it. I just read the quotations. It's not connected. Okay, yeah. So all I was going to read was that Don't worry. I was going to give you a quotation from the great American physical chemist Robert Mulliken, who said, "I wish to emphasize my belief that the era of computing chemists, when hundreds, if not thousands of chemists will go to the computing machine instead of the laboratory, is already at hand. There is only one obstacle, namely, that somebody must pay for the computing time." Well, I want simply to pay a tribute to Arieh and say that his work provoked me 10 years ago into a new line of inquiry, and our results have been stimulating. We do not always agree with Arieh, but we follow in his wake, and we look forward enormously to his presentation today, and his title is not even on the sheet. His title is Recent Results in Computation of Biological Mechanisms. Arieh, you are most welcome.
Okay, it is a great pleasure to be here. I would like to thank Mike for the introduction and, of course, for the invitation.
This is my computer. Yeah.
Okay, so I will try to describe some of our studies of the nature and the origin of enzyme catalysis And as a general introduction, why you need to study it, there are cellular processes that if you understand them, this is signal transduction, one of them in a cell. It looks and it is very complicated, but if you want to study it on molecular level, one notes that all of these are molecules, and in principle, you could study some of them one by one, and then the communication between them. A lot of these life processes are managed by enzymes, and I was surprised how many people don't know what is an enzyme. So enzyme is a protein molecule that bind substrate chemical entities and then makes them react faster than without the protein. Here is an enzyme doing a reaction, and the same reaction without the enzyme takes very long time. So one would like to understand why the enzyme works so much faster. This is about a 100-year-old question. And also how to manipulate enzymes. There are a lot of questions, and you could crystallize this question without solving it by saying that in case of reaction without the enzyme, you have a high activation barrier. It takes a long time to break the bond. And somehow the enzyme makes the time of breaking the bond faster. So the question is, of course, for quite a long time, what is about the enzyme that makes it so effective? And although there has been enormous experimental progress, and of course structural studies that without them we will never understand enzymes, I would argue that without computer or computers, you cannot really get a definite answer. Even with computer, it's difficult to do so, because it's very hard to look on the forces within the enzyme and to figure out what they are experimentally. And while mutation experiments are very revealing, they still don't give you a unique answer. So you want to use computer, but the question is how? Now, part of this problem has been solved quite a long time ago. You could represent a molecule. Yes, you could represent a molecule as ball and sticks and get, ball and springs, and get the behavior of the spring to correspond to the actual molecular forces. It's not okay, but fine. This is supposed to move. But you get the idea. You could represent the bending of the angle, the stretching of the bond, and this approach, which is called molecular force field, has been very instrumental in describing medium-sized molecules and also large molecules. Some of the work that was done in our group, this is Schneur Lifson, Mike Levitt, when he was very young, and me. And these are some of the works that develop force fields that have been used for protein to describe their dynamics and so on. Now, the problem is to describe the chemistry, because you cannot describe bond breaking by ball and stick. So you could stretch a bond with springs, but you cannot break it, because when you break a bond chemically, electrons are moving, and you have to do something more. Essentially, to describe the motion of the electrons, you have to use quantum mechanics. So the issue is how you describe the quantum chemistry of bond breaking while also taking into account the very large enzyme with the many atoms, which for a very long time you could not put all of them on the computer, and even now it's not so useful. So a solution to this, what is called the QMMM, which is quantum mechanical molecular mechanics. I don't know why we don't see it here. And in this approach, you describe the reactive part quantum mechanically, and the rest of the enzyme and solution you describe by force fields, where you take into account the residual charge of the water molecules, the protein polar groups, and so on. Now, it's easier to suggest such approach than to make it quantitative. For the few theoretical-oriented people, we try different level of approaches, and the one that seems to be the most quantitative one is an almost high school approach called the empirical valence bond that looks like this. If you want to describe a reaction where acid attack carbon and release Cl, you could describe by force field the reactant and products, and then mix them by mixing term to get the reality. And the reality in this case will be the chemistry without the enzyme but in solution. So you train this approach to tell you about chemistry in solution, which many times you know experimentally, sometimes you don't, and I will discuss it. And then you replace the solution by the enzyme, so it's a comparative approach, and get, in fact, quantitative information on enzyme reaction and enzyme catalysis. Now, for people who believe only in high-level quantum mechanics, we, during the year, design approach that we call paradynamics, where you use a simple approach, the EVB, for getting the free energy profile, and then you do perturbation from the product or transition state to ab initio, and from reactant to ab initio. I show it basically just to tell you that this approach is okay even for orthodox ab initio people. So now we have this approach. This is an example of studying, this does not work too well. This is an example of studying dehalogenase. This is the reaction in water. This is in the protein. And we could get the same reaction by the EVB and by then going to ab initio. So we have an approach that seems to be reliable. It calculates free energy profile, not potential energy. So it includes entropy and other effects. Now, we have used this approach for many years, and it could be used to study basically any type of enzymatic reaction. Some of them go back to, let's say, G proteins, which are very interesting. And the example, we have many examples. This is, for example, the elongation step in the ribosome, where you add extra amino acid to the elongating peptide chain. This is the messenger RNA. And once this group, which is called elongation factor, come to interact with the messenger RNA, the GTP here become very active, which means that EFTU become catalytic, and the reaction becomes very fast. And there is a question, why or how could you transfer the information from very far away to the active site? Now, this has been controversial. This is from work of Ramakrishnan. Some people claim that the histidine, which is brought near the phosphate in the active form, is being used as a base, but we have shown long ago in study of rust that this is not the case in related protein. So you want to solve this. Unfortunately, Ramakrishnan gave, this is really annoying, Ramakrishnan gave the structure in the active and inactive form, so you want to convert this X-ray structure to chemical information. Now, it's not simple. You have a reaction, phosphate hydrolysis, with very high energy transition state, and it's very hard experimentally to get the unique answer. There are very many reasons for this. One, it has been controversial whether the reaction is associative or dissociative for a very long time. And we show in several papers that it's very hard experimentally to really determine it. To add pain to injury, sorry, this is showing how hard it is to decide whether it is associative and dissociative. This is, I'm sure there is a Russian version to this cartoon. These people, one is blind and the other is apparently also blind, and they touch different parts and reach different conclusions. Now, recently, it appears to be even more complicated, whether there are one water or two water. Really hard to figure out, and we felt that only high-level calculations will tell you the answer. And we done QMMM in solution, very extensive calculation with ab initio QM part. What you see here, could we have another? Okay, I will use this. What you see here, great. Okay. Okay. So there is magnesium here and some quantum water, and the conclusion was that in solution, it's actually two-water mechanism. And with type of complicated surface, we fit them to this simple EVB and start to study the reaction in different systems. This is rust, but it's always the same movie. What you see here, no, it's, you have to touch this. It's okay. Okay. Anyhow, there is a nice movie here where this water molecule transfer proton to the phosphate. This is the magnesium, and you have a reaction. We could try one more time, but I don't think it will work somehow. What? Does not matter. You could see it on my site. The main point is really not the movies. We could do a lot of them. The main thing that you get quantitative description of going from the chemistry in water to the chemistry in this protein. And overall, we concluded that the reaction is basically working in an allosteric way where the long distance effect change parts like the P loop and also the histidine. This is structural change. This is chemistry. This is in the native. This is in one of the mutant. And similar story on similar enzymes. Okay, now a lot of these studies were summarized in a very big review, Why Nature Chose Phosphate. And in similar way, we study many interesting systems, and it's partially relevant to medicine, partially to general interest. So now we do a small switch in gear and try to ask, well, we understand we could model different enzymes, but why enzymes work so much faster? And there are like 30 suggestions of why enzymes work, going at least from the 60s. And the advantage of a computer is that you actually could check different hypotheses and see how much this suggestion contributes. And this is, some of you know it, this is Linus Pauling in a famous lecture where he was supposed to explain how enzymes work. There were no structure of enzymes. Basically, he pointed out that it's change in free energy. He did not even say it's transition state stabilization. So the question became really open. And in our many studies, these are reactions of different enzymes in water, and this is in enzymes. And in any case that we touch, it appears that the change in barrier is due to electrostatic effect. Now, this is a little strange, and the question is, what type of electrostatic effect? And it's not trivial and sometimes confusing. Essentially, next week you will see a short letter in Proteins where I attack somebody who tried to explain what we said. So I have the proof now. So this is a reaction. Maybe I will first show you a schematic picture. This is typical reaction in water, where you move charge from one group to another. And when this happens, and here is the transition state that you want to push down, what you find is that during this reaction, the water molecule have to change structure. And this costs energy called reorganization energy. The larger is the reorganization energy, the higher is the barrier. Now, the enzyme realizes it, and instead of allowing the equivalent of the water molecule to rotate, it had its polar groups already pointing to the right direction. So the reorganization energy is smaller, and the barrier is smaller. You could see it here, where this is the reaction in water. You move to delocalize minus-minus transition state, and you pay by rotating the water. This is the same story in the protein, where you don't have to rearrange the polar groups. Now, one of the confusing issues here, which is the subject of the letter we have in Proteins, is that the interaction energy between the surrounding water and the transition state and the surrounding enzyme in the transition state is the same, which means that the catalysis is not due to the interaction energy. It's really due to the fact that you have to pay energy in rotating the environment from ground state to transition state. And therefore, this is where the catalysis is located, and maybe I will have a slide that show it. And so now, sound very nice, very confusing, but a way to address it is to ask why other proposals are not so correct. So you could go through textbooks, and I guess I have to get a better picture. This is one of the most famous proposals, the desolvation idea, belong to the family of ground state destabilization. It sounds very logical. You have a charged substrate. It's very happy in water. You move it to the enzyme where he's supposed to be surrounded by hydrophobic environment, which I will tell you in a minute is incorrect. And therefore, you have to climb less to reach the transition state. Another version of this proposal is that you have a charged substrate and you place it in an enzyme where you have the same charge. So again, this guy is unstable and you have a faster reaction because you are already closer to the transition state. In general, this ground state destabilization do not work when you check them by computer. And I could tell you why, but we'll just show you a very nice example. So the example is one of the most proficient enzyme, this orotodine decarboxylase, which was studied by Wolfenden, and it releases carboxylase, CO2, when it goes to the enzyme much, much, much faster than in solution. So originally it was argued that the active site is non-polar, like the dissolution idea. Then when the structure was solved, as we predicted, it appeared to be very polar, with negative group just near this negative group. So again, the argument was that, well, this enzyme works so fast because minus repel minus. Now, of course, if you think about it, if this minus was unstable, it would pick a proton. But of course, did not stop people who suggested it from proposing it. So eventually, we done calculation. We show that actually, we have here an ion pair of a lysine from the protein and this carboxylate. And the aspartic stabilize the lysine. And when you go to the transition state, you have a larger dipole, which benefit more from the pre-organization. But the nicest thing, which would be the easiest to understand, came from a set of experiments by John Richards, which meant to show that this idea is incorrect, that actually the enzyme work by what is called using binding energy from distant group to put the unstable chemical part in its environment. So the idea was, this is the orotodine, it's connected to a phosphate, and if you have unstable carboxylate here, you compensate for it for binding a phosphate far away. So the idea was to cut this bond, and once you cut this bond, the enzyme will become, this guy will become stable, and there will be no catalysis. So they did experiment, they cut the phosphate group, and in fact, the enzyme become very poor catalyst, which support this usage of binding energy. But they done one too many experiment and put again inorganic phosphate in the solution, and the enzyme catalyzes again. So if you think about it, it shows that the idea of, at least here, of using binding energy to push parts of the substrate to unstable environment does not work here. And the same experiment were done with triphosphatizomerase and other systems, and they lead to the same conclusions. So I will give you one more detailed experiment. There is this ketosteroid isomerase, again, very efficient enzyme, about 11 kilocalorie catalysis. It has this steroid with CO that originally people felt that it's still keto, but actually it goes to enolate in the enzyme. And the argument would be that these two guys form oxyanion holes that stabilize minus here. But Dan Hirschlag at Stanford was obsessed showing that it's not electrostatic. He did a set of very nice experiments. He replaced the steroid by a substituted benzene, where changing the substituent changed the charge on this oxygen. And he showed very nicely experimentally that changing the charge does not change the binding energy. And therefore, the simple conclusion is that electrostatic is not important here. Of course, we did not agree with it, so we tried to do the calculation of generating the charge in the transition state in water and in solution. In this calculation, you are gradually form larger and larger charge and calculate the free energy. So with the steroid, we got all the catalytic effect. However, with the phenolate type systems, we got the same result in water, dash line, and in the protein. So you try to think this is the computer result. With computer, you could find what it is due to. And what you find is a very nice story, that when you have this phenolate, when you have it in the uncharged form, it could be in several directions. In the charged form, the transition state, it's the same as with the steroid. Exactly as my story on water, in the transition state, the interaction is the same. But at the reactant state, the steroid is fixed. This one is not. Now I'll show you something which is in the paper. It's very hard to follow, but it's very instructive. When you have a reaction in water and you turn charge on, you could do it in two steps. First, turning the charge on and keeping the water. Then with the charge, you rotate the water. So you have two terms. One is the average potential energy of forming the charge where the water is in the initial state, and the other is rotating the water when the charge is on. When you do this term of rotating water when the charge is on, gives you a lot of energy. But when you do it in the protein, you gain nothing from rotation because the polar groups are already pointing in the right direction. So the catalysis comes essentially from this very strange term, the interaction of the solvent that does not know about the charge. So it's this one, I was wrong. The interaction of the solvent that does not know about the charge and is zero in water with the formed charge. You should see it in the paper. Now, the nice thing, again, this is not experiment, is that Steve Boxer done a series of experiments on it. And eventually, he concluded and kind of proved that it is electrostatic by looking on vibrational Stark effect, calibrating it on the, basically, groups around it and getting the whole catalysis with different mutants. So now, how much time I have? 10. Okay, so we could give you the same story about dynamics to show you that dynamics does not contribute to catalysis. We could show you, it's a nice story, but since I talked too slowly, I did not come to it. We could tell you the same story about why tunneling, maybe I will tell you, why tunneling, which is by entropy, does not help to catalysis. Again, nice story. And I will give you only one more and then move to another subject very briefly. So it's very nice to claim that enzymes work by tunneling, by compressing the distance between the reactant and product and making the reaction faster. And there was article by Ball in Nature called by design or by chance about tunneling. And the nice thing is that you could calculate tunneling in enzyme by path integral, where you replace the light particles by beads of quasi-particles. And you could calculate the correction of the free energy due to quantum mechanical tunneling. And we've done a lot of this. Johan Aquist did a lot. And what you find is that you have the same tunneling effect. Tunneling exists, but it's the same in water and protein. And the nicest thing, which you should think about it next time when you teach this, is that the original proposal is that the enzyme makes a very narrow barrier, and therefore hydride or hydrogen could pass faster. There is only one deadly problem here, and this is that when you push closer acceptor and donor for hydrogen, instead of having a narrower barrier, the barrier basically disappears. It's something that is completely ignored by the tunneling community, and essentially the tunneling becomes larger when you pull the group apart rather than compress them. So we wrote some article where we took a tunnel in Slovenia and make the point that you cannot tunnel when there is nothing to tunnel through. It's on the cover of the journal. So my main point, you could check different suggestions and see if they're right or wrong, and still nobody would believe you unless you could design enzyme. So 5 minutes on enzyme design. You want to be able to take enzyme, to change the amino acids, and to get a better enzyme, which is much easier to say than to do. This is, for example, Christmas mutase, where we took different non-mutants and shows that we could reproduce them. Yes, this is Christmas mutase. We could predict quite well the effect of non-mutation. This is the allogenase, again, not so difficult to do it. It does not take too much computer time. So we try with this idea to go into camp eliminase, which was one of the most highly publicized enzyme design. And by the way, none of these enzyme design experiments really produce anything. The only things that produce it were experiments in directed evolution. Nothing was produced by the computation, just to make you sure about it. So we try to study the non-directed evolution, and seems to get a nice correlation, which look promising between the calculated and observed result. Now, we try to understand why this is happening, and found something really annoying. We found that the directed evolution, which does not take the time of evolution, it's rather stupid. It does what I told you that enzyme never does. It does accelerate the reaction very little by dissolution. You could see it here in one of them. In the native, there are more water molecules, which mean that the base, the glutamic acid, is more stabilized in the native. In the mutant, there are less water molecules, so it's less polar, and the acid becomes less stable. Of course, there is a problem. There is a limit. After some limit, the acid will become protonated. Now, we felt very unhappy with this. And of course, people say, look, here evolution get you ground state destabilization. And our first argument was that this is not really evolution. We don't know how long you need to do it. And fortunately, Don Hilbert, the original experiments were done by Dan Taufik in the Weizmann Institute. Don Hilbert continued, and recently he suddenly found a faster enzyme. Nothing remarkable, but this enzyme appeared to work by transition state stabilization. It managed to find a way to stabilize the oxygen. Maybe we'll have a better paper. So anyhow, Don Hilbert found a way to get it. So we first done, in a minute there will be a very interesting conclusion, so I will still pull until it. We showed that we could reproduce this chain of directed evolution, which was very encouraging. But it was, we've done some computer experiment. We tried to do better and we keep failing. And then we look on this series of directed evolution, and we were trying to find a way to have pre-organized environment to the oxygen of the camp compound. And we found that only at the end of the directed evolution, a single mutant creates pre-organized environment. If you go too backward, where you already have the right dipoles, they do not create oxyanion hole. Now, in other words, we found something very, very disappointing for people who try to do rational enzyme design, that it would not work with a single mutant. You always need few other mutants to help you to put the critical groups in the right direction. This explains why we could very easily take known enzyme and destroy it on the computer. But up to now, we did not succeed to create a dramatically better enzyme because we need much more combinatorial tricks, and we are not there yet. So this is the same story. I had a long story on drug resistance, which I will not tell you. And the conclusions are that enzyme work by pre-organized environment that does not exist in solution. This I will save you. And in general, we found in all of our studies that electrostatic is the key for structure-function correlation. These are people who contribute to this and other works, and here you see them better. And thank you very much.
Do you mind questions? You don't mind?
Of course not.
You don't?
I thought you asked them.
We have a few minutes for discussion. If there are questions, they're welcome. And microphones are over there. There are at least 3 microphones ready to work. You have a question? А можно помочь с микрофоном? Microphone is coming.
Thank you, Arieh, for this exciting lecture. And my question is, now with all your experience in understanding enzymes, what do you think still can be improved in pre-organization and in some other steps of catalysis? And how much?
Okay, the pre-organization which is done by evolution cannot be improved too much because it's not a completely fixed enzyme. So I don't think it, with perfect enzymes, with enzymes that done the maximum that they can, I'm not sure how much, but compared to directed evolution, there is huge place for improvement. I'm not sure it's answered the question, but...
Okay, thank you.
Arieh, I have a couple of questions. Is this alive? So the first one is you ended up talking about tunneling. This is a fascinating problem. And one question that's been put to me recently is there a difference between tunneling for a hydride and for a proton?
The answer is that the magnitude of the tunneling, in a complete contrast to all, usually the tunneling community, increase when you pull away the donor and acceptor, rather than compressing them. And this is shown in the following picture. Just 1 second. What happened in all reaction of atom transfer is that when you push them apart, these parabolas go away and the barrier goes up and it becomes narrower. So essentially there are molecules. I mean, it's very hard to find it in regular organic molecule, but if you look on them, some of them, Mavri wrote a series of them, which involve hydride transfer. So there is an issue of when the distance is larger with hydride or proton transfer, and it could be that in hydride it's larger. Now, the enzyme, I don't think, care about it too much. So the answer is that the quantum mechanical, if you have the same surface, the quantum mechanical effect will be the same, unless somebody will try to describe the hydride as a three-state couple proton transfer, hydrogen transfer, and then it will confuse the audience.
So my second question is, you gave us the wonderful panorama at the beginning of the complexity of biology, opening up as structural studies drive deeper into it. And it reminded me that I went to a meeting in London 50 years ago when Michael Dewar proposed that the limit of computing application to chemistry and biology was within reach because of the speed of the electron down a copper wire. And we know how wrong he was.
Yes.
But now you drive us into an area where you can look at 100 molecules, 100 heavy atoms, yeah? But then,
No, I could look at 10 million, but okay.
Then we have Venki Ramakrishnan now looking by cryo-EM at enormous assemblies. So my question is, how is computing science going to advance into this new zone where we are 100 times larger in the molecules we are exploring?
Okay, so we have, this is the part that I did not give, or one of the part, where we have multi-scale modeling of the ribosome and translocon. So we do it by some simplification. And I don't see, I mean, we do myosin, I mean, you have to use the right question. We have a project where the leader of the project says that he wants to model the whole cell by atomistic simulation, and we had a workshop, and he asked how many years, and some people say 3 years, and I wrote 75 years until this will work. But you could use simpler approach to ask the same question. So computers with the right approach could ask questions on very complex systems even now. And with the all-atom approach, probably they will never be able to do it.
Okay, it's time to move further. Thank you very much again.
Welcome.
got a title? Is there a title here for Sasha? It's not in here. Yeah, combinatorial approaches. So it's my pleasure now, ladies and gentlemen, to introduce the second speaker of the morning, Professor Alexander Gabibov. As we all know, there is a strict protocol in science that you are not allowed to referee manuscripts that are submitted by somebody with whom you have recent research collaboration and publication. Fortunately, that does not apply to being a chairman, because Sasha and I share many publications together and many enterprises. But I'm reminded of a story of Lord Kelvin. They once said to Lord Kelvin, he was J.J. Thomson, Dr. Thomson, you're a lucky man. You ride on the crest of the wave. And he turned around and looked at this young man with a very broad Scottish accent and said, but you forget, laddie, who invented the wave? And in a sense, then, I look at the work of Sasha Gabibov in this institute over the last 15, 20 years and think that he rode the wave at the interface between chemistry and immunology. And I won't say he invented this wave, but this business of bringing together chemistry and biology and dissolving the interface has been a revolution in all the countries, East and West. And I'm so pleased then to introduce Sasha to give us his lecture this morning. Sasha, the floor is yours.
Thank you, Mike. It's really a big exaggeration, because we remember very well the science paper of Richard Lerner, Penkovic, and Schultz, I guess, in '91, when they say they're on crossroad of chemistry and immunology. You are true that I will speak a little bit, I will talk a little bit about my old story of antibody, and I must apologize, my presentation will be a little bit eclectic, because I want to show some approach which we are doing in my lab here during these last 25 years, before I worked in the Engelgard Institute. And all these approaches may be regarded, regarded on the vector of combinatorial approach. So I will give you some examples, which antibodies will be the first. And this talk will be done from two affiliations of Chinnikov Shemyakin Institute, we are here, and Lomonosov will be head of the department. So, but I have a big pleasure to say that this institute during 30 years is guided by our famous chairperson, Professor Vadim Ivanov. And it's my pleasure. He is absolutely against any celebration, but today, by chance, he has 80-year-old, his anniversary, and I took my time from my mic. Please look on the slide, yes. I take some time from my scientific part to say from all our institute, all groups, all colleagues, all members of Russian Academy, foreign members of Russian academies, Joseph Schlesinger, Nobel laureate, and other invited speakers, how we respect Professor Ivanov's work in peptide chemistry, in mass spectrometry of peptides and peptidomics generally. And I have a little present to him from our institute. He's not a hard drinker, but I think a bottle of this champagne will just good for this day. Marina will say also a few words. We have some telegrams from Academy full members of Academy, Professor Gliko, from the biological department of Moscow State University, academician Kirpichnikov, she will go here, from our brother institute, very close to our Engelgard Institute, Director Alexander Makarov. So I think we have a lot of telegram, but Professor Ivanov insisted that we know that it's a birthday, but we are not have some official celebrations. So as it was mentioned on the main talk, maybe I will cut some slides, but I say what is biodiversity, and I will try to to take little information on three main topics in which we are doing in my lab. And I have the privilege to say that a couple of my ex-student postdoc, now independent young researchers, will have their own talks, and they will give more information what we are doing more. But certainly first part will be covered by in silico maturation of antibody. My friend, classical immunologist like Monsef Zouali, B-cell, one of the B-cell leader maybe will be against, but will show the results. Microfluidics, how we screen the platform, because it's, in the case of combinatorial approach, it's very important to have a good screening approaches. I think it's a revolution thing. And also I will say something about general thing about library, how we will use it in different biomedical approach. So I will start with some trivial messages, but I have to do it because not all are familiar with this field. So I think that combinatorial chemistry and biology is a real hallmark of 21st century. It made a revolution in this approach. So I also have a very close concern, which Arieh say about direct evolution. But anyway, this approach made a very big, how say, achievement.